trigger, right? You know that this is going to be a paracrine, and since this is a neuron releasing it, we'll call it a neurocrine. You can think about neurocrines in two different light, although we tend to just group them together and call them neurotransmitters. But if the response is rapid in the postsynaptic cell, that would be technically called a neurotransmitter. If the response in the postsynaptic cell is a little bit slower, probably because what that triggered was a second messenger system, it often gets called a neuromodulator. Modulators don't work as quickly, but you will see, I mean, I group these together, and nearly everybody groups these together, that if a neurotransmitter or a chemical is released by a neuron, we're going to call it a neurotransmitter. Okay. Norepinephrine works by a GPCR mechanism, but yet you'll hear everybody talk about norepinephrine being a neurotransmitter. Okay. So keep that in mind. Neuromodulator, neurotransmitter, kind of the same thing, okay? But the responses that they cause once they bind to the receptors can either be fast or slow. Fast responses work by them just binding to a receptor that's linked to an ion channel. Very quick change in this cell, right? Obviously, you're aware of the fact that if we have to work through GPCR receptors here, right, that is going to take a little bit longer for that to occur because we have this signal transduction pathway that has to occur. The benefit of these, though, right, is because it's a cascade, we have more opportunity for regulation. This, you either get it or you don't because it's only a single binding, right? This one we can regulate along the way through that cascade. And it activates a second messenger system. What the second messenger pathway does, it can open and close ions and create ion flux, or it can modify existing proteins if it's linked to a kinase or something like that. So it really just depends on what we're going to get. But these are the responses that we're going to see. And when you get a response in the postsynaptic cell, if it is a gland, that would result in either the gland secreting something or not secreting something that it produced. If it's a muscle cell, that muscle cell membrane is going to be depolarized also, right? So neurons can generate action potentials, and so can sarcolemmas, right? So smooth muscle cells, cardiac muscle cells, skeletal muscle cells, they all have the ability to generate action potentials just like neurons. What that means is they must also have those voltage-gated sodium and potassium channels, right? And they create action potential spikes. What's a little bit different is that resting membrane potentials are a little bit different in different cells. Threshold values are different in different cells, and the spike is different in different cells. So these gates open and close at different voltages, but you still get that same kind of pattern like that. This might be plus 20 instead of plus 30. The threshold might be minus 60 instead of minus 55 or minus 40. The resting membrane potential might be minus 90 instead of minus 70. It depends on the cells we're talking about, and we'll see those in context here in a little bit. But the point is that you still have that sequential activation gate opens and activation gate closes, potassium gate fully opens, sodium influx precedes potassium efflux, right? You get this kind of wave of depolarization that spreads down the cell membrane. What do muscle cells do in response to that depolarization? They contract. That's one of the things we want to talk about later today is how does that electrical signal on a sarcolemma trigger muscle contraction? So we'll be looking at the interactions, some special organelles that are inside muscle cells called myofibrils, and they have these units in them called, in striating muscle, sarcomeres. And that allows for myofilaments, actin and myosin, among others, to interact with each other and generate tension. And so that's what we're going to talk about a little bit later on. So that's kind of our goal today, is to kind of look at that. Now, one thing that you have to appreciate is that if we're talking about, well, let's think of it this way. We know that what's important in how the brain functions. We're not going to do a lot with the central nervous system because we'll actually see it in context throughout the rest of the term as in different parts of it regulate different parts of the body. One thing that's important is that what makes the central nervous system work is that there is a unique sequence of neurons that are hooked together, right? And that's really how things work in the central nervous system, is the fact that we create these neuronal networks and these neuronal pathways. And it is these specific pathways that will give us a particular response, right? Allow us to associate things, to integrate things, or whatever is the production of it. So what's important is not really what the neuron is, and that the neuron can generate an action potential, but what neurons are working together in a pathway. Anytime you learn something, so you didn't know how to do something before, even right now while you're learning and you're trying to understand these things, remember the significance of chemical synapses is that we can break and reform them. What allows us to learn something is that we create a unique network of neurons, a pathway in the brain. That's learning to do something, is creating that network. And these neurons are breaking and reforming. We are releasing neurotransmitters into synapses that are generating this kind of flow of information around different parts in our brain. I mean, that's an oversimplistic view, but that's really what's happening, is when you learn to do something, you have created a pathway that wasn't there before. Or maybe the pathway was there before, but it wasn't being used, meaning you weren't releasing certain neurotransmitters into the synapses between these neurons. Memory is the ability to maintain and recall and reuse a pathway, right? Obviously, you know you create pathways and you learn something. And the more you use the pathway, the brain should think the more important that pathway is, right? It should reinforce that pathway. And what you reinforce when you do memories is you reinforce a synapse, right? I mean, obviously, the most reinforced synapse would be, wow, this is so important, let's make it electrical synapse, right?